know anything specific. I'm just ready. Those are Googling. By Googling
instead of like dog eating. It says leave it there.
Just leave that there. I'll make sure.
Hi everyone, I guess we're going to get started here to allow Dr. Richard some time for his presentation. Dr. Kinahan couldn't be here today, so we asked him to step in to introduce our illustrious next speaker, so I'm very happy to do that. For everyone probably knows Dr. Richards. He's been with the department for 34 years now and is looking towards retirement at the end of the month, so we're really sad to lose him, but we're happy he'll still be local and we can reach out. He said he's available for questions anytime. So I just wanted to give a little background for anyone who doesn't know Dr. Richards, that he's been really a pioneer in our field for developing advanced MRI techniques and for doing translational research studies in multiple areas directed towards brain imaging. These include MR spectroscopy, functional MRI, I have my little list here, diffusion MRI, and even incorporating EEG signaling into the MR experiment. He's had many years of highly productive grant-funded research, both from NIH funding and other sources, and resulted in well over 150, I think it's something like 175 papers I found when I looked online, and that was not a very official search, so I'm sure there's more. Highly impactful publications, so just really amazing work coming from all of those studies. He's played a key role in multiple clinical trials to test new treatments for brain imaging, and I'm hoping he'll show us some of the exciting results of that today. So personally for me and others here at UW, he's just been a wealth of information and a very generous source of someone we can approach anytime questions arise from our MR acquisitions or post-processing needs. And so just really appreciated his generosity to make time for us for that and also for brainstorming new ideas. So I have a few notes from some former colleagues or current and former colleagues and collaborators that couldn't be here today. Maybe you'll recognize some of those names. But one is Satoshi Minashima, who's currently at the University of Utah, who worked previously with Todd in several ways. He reached out to give you congratulations. He said you've been a visionary in MRI research and a trendsetter. And I still remember your PEP-C protocol implemented on 1.5T and the great results that came out from that, that research. You're fun to talk with, and he wants to know if you're still rollerblading. He says he hopes the retirement will give you more time to focus on you and your family, and he hopes that you will come to visit Utah. Stefan Posse from the University of New Mexico, who I guess has worked with you here as well. Yes, he said that he remembers some late nights collecting PEP-C spectroscopy data, and he really appreciated your help with trying to figure out how to manage all this large data that was difficult to deal with in those days. And he just went on to appreciate your work after that, using evaluating metabolic changes with language activation, and he just wants you to wish you the best on your retirement. Dennis Shaw, more local, couldn't be here today also, but he pointed out that he remembers losing a basketball game when he started out as a resident to the faculty, and you were on the team in those days. So he remembers losing the basketball games, and also one of the first projects he worked with you was a monkey model of ethanol exposure, which he refers to more as the drunk monkey project, which he he had a lot of fun working with you on. Stephen Dagger, I'll just end with one quote from him. He couldn't be here today either, but he said, "Todd, you were my guide and protector, venturing into the realm of MR. I secretly, maybe not so secretly, thought of you as Gandalf the Wizard. Even if you didn't have a beard and were infinitely less taciturn. I will miss you."
Thank you.
So with that, I want to end and invite you up to regale us with all of your interesting.
Okay, I'm glad to be here, and I'm going to go over briefly some of the results we've had over the years. And so there you go. So when I first came to the University of Washington, I had mentors who helped me obtain my first grant. And if you're a new investigator, there's concepts for a young investigator. One is publish or perish, or get a grant or perish. So these are important concepts. And my old advisor, Thomas Bunninger, was one who really helped me, and two other people, Ellsworth Alvord, who's passed away a few years ago, and Kenneth Crone, who recently retired. So all these people helped me to get my first NIH grant, and I appreciate their help over the years. Okay, so first of all, I'm going to talk about MR spectroscopy. So spectroscopy is a technique for non-invasively measuring chemicals in the body, and this technique can be performed on most scanners. And MRS can't measure all chemicals, but you're limited to a certain set of chemicals, and I'm going to go over those. There's a certain set of chemicals that we can measure with spectroscopy. So why is MRS important in the brain? Because the brain has a delicate balance of neurotransmitters and chemicals, so if this balance is disrupted, it could lead to different diseases. So I studied multiple sclerosis for quite a few years, and in that disease, we showed that there was a very good clinical correlation between symptoms and certain chemicals that we can measure. So one of the findings that we showed is that normal appearing white matter on MRI can have pretty large chemical abnormalities that you wouldn't be able to detect unless you did MRS. So anyway, MRS is very sensitive. Okay, a history of the development. In 1983, and from Yale, Schulman's group and our group at UC Berkeley were among the first to do proton spectroscopy of the brain. We were the first. So we did proton spectroscopy of the brain. Before that, they had done a lot of phosphorus. So it's interesting. Phosphorus came before proton and spectroscopy in the brain. So they had done a lot of phosphorus spectroscopy in the brain, and I'll show you some slides of that. But in 1984, Bottomley invented the press sequence for localization. In 1989, Fromm developed the STEAM sequence. In 1990s, we and others developed some techniques for multiple voxel imaging, and then in the 2000s, there's been an explosion of new papers on the use of spectroscopy. So it's developed over the years, and there's some groups that have really been able to do a lot with it. So there's a few good books on spectroscopy. One is called Biomedical Magnetic Resonance by Thomas James and Margulis. I knew both these people at UC San Francisco. So before I came here, I did my postdoc at UC San Francisco, where I knew both of these people, Thomas James and Margulis, who were great mentors. Here's another good book on spectroscopy. And by the way, if any of you want a copy of these slides, I'm happy to send you a copy of all these slides. So spectroscopy, to learn it, the principles and techniques, there's a good book by DeGraff. Okay, now just a few concepts. Whenever you put body, tissue, anything, a magnetic field, it gets transformed. Okay, we have a transformation. Whenever you place any tissue or any kind of material that has spins, it gets transformed from this regular state into a state where there's different energy levels. So you go from a normal state and then you put it in a magnetic field, voila, it all of a sudden has these different energy levels. And in spectroscopy, it's especially important because the different molecules will have different states depending on their molecular structure. So this electron current causes the magnetic field to change within the molecule and give rise to these different peaks that I'm going to show you. So in spectroscopy, we have these different peaks. We have the amplitude and line width. So these peaks all have meaning, and the different chemicals have different frequencies. So the different chemicals you can measure with spectroscopy, and they have different coupling constants. I'm going to go over that. So for example, ethanol. So I don't drink alcohol. But I don't drink. But ethanol is a good example of a molecule where you can measure all of the properties of spectroscopy. So ethanol has three proton groups. A CH3 group has a place that is represented. So the CH3 group sits here in frequency and is split into 3 peaks. The CH2 group sits right here and is split into 4 peaks. And the OH group sits right here in the frequency. So on the x-axis is frequency. On the y-axis is amplitude. So when you take an MR spectrum, you can measure the properties of these different molecules based on their splitting and their frequency. So I'll show you as it becomes more important. So ethanol is a good molecule to learn spectroscopy because of its splitting and the way that, so these carbons here, they interact with each other through their protons, so that this proton can look over here at this proton and say, okay, I'm interacting, I'm going to split. So it's an interesting mechanism of how these protons interact with one another. Okay, the other important part about MR is we have to do the Fourier transform. So my advisor, my thesis advisor, he taught this course on signal processing, and I got my first software to do the Fourier transform. I was so happy. I can now do a Fourier transform with this software that I've used for 35 years. So I've used this ability to do Fourier transforms given to me by my old advisor. So this is written in Fortran, the ancient language of, Dead Sea Scrolls, Fortran. So it comes from, Fortran is a really good way. But anyway, it still works. A lot of people now like to use Python or whatever. Python works fine. But to do the Fourier transform is a very important part of MR because we have to convert the time domain to a frequency domain. And you do that using the Fourier transform. And when you get this FID signal that comes out of the scanner, and you have to do the Fourier transform, and sometimes I have to do a 3-dimensional Fourier transform because in each dimension it needs a different conversion from time domain to the frequency domain. So I do all these Fourier transforms, and in this slide it shows a good example of different frequencies and how it looks when you do the Fourier transform. And so here's one of our early papers that we did. These people are from UC San Francisco. So Craig Davis, Bruce Barker, Burnett, and Jeanette are all from UC San Francisco. And we were doing a lipid water ratio of bone marrow using spectroscopy. Okay, and then later on, we also did spectroscopy with all these characters here. There's James Bowen, Ellsworth Albert, Kenneth Maravella, Steve Dager, Lynn Rhodes, and Stephan Posse. So here's a paper that we wrote on basic concepts and emphasis on multiple sclerosis. So I studied multiple sclerosis for many years, and we would do spectroscopy on these patients before and after they got some kind of a treatment. So I always enjoy pairing up with a clinician who wants to investigate new treatments. And so they'll come to me and they say, Todd, what's the best MR signal that we can use to compare our patients before and after they get their treatment? So I make suggestions on different MR parameters that you can use to test these treatments. This is just a diagram showing a one-dimensional way to divide up the signal. So we've come a long way since just doing one dimension. We can do a lot more than one dimension. And now this is an important pulse sequence that shows you that you can phase encode. So we have a 90 and a 180 degree pulse, and then in between we have a phase encoded gradient, and then we have an echo over here, and we can use that pulse sequence to get spatial information from the spins, but also get chemical information. So you can get both spatial information and chemical information from the same signal by using this kind of a pulse sequence. Okay, now this is actually our daughter, Carolyn. She was a willing subject, very willing. She's laying here in the scanner. She's about 8 years old. Is that right? 8 years old. So, yeah. So anyway, another important part of this photograph is this head coil. This head coil was developed by Cecil Hayes, Mark Mathis, and this head coil was used for many years. And it was just so important to get the best signal from the head using this special developed head coil developed by Cecil Hayes and Mark Mathis. So we got really good signal from this coil, and we were able to do lots of projects with that setup. Okay, and then we did lactate imaging using MR spectroscopy. So you can do lactate imaging using different techniques. And lactate has, I'll show you why it's important in the brain. And I'm going to show you a paper. Okay, here's the structure of lactate. It's got 3 carbons. It's got 2 OH groups and a double bond over here. And it's got a CH3 group over here. And that gives rise to a certain frequency and splitting that we were able to use to separate out from different chemicals that are in the brain. Okay, so before I get to one of our favorite lactate papers, I'm going to show you, here's some more chemicals we can measure in the brain. We can measure N-acetyl aspartate, creatine, choline, myoinositol. All these chemicals are important in the brain. And then here's some glutamate, glutamine, glucose, lactate. So these chemicals have a different spectrum. So over on the right, over here we have the spectrum of NAA. So one way to calibrate is to take a small amount of the chemical, put it in a test tube, and scan it. So you can take the chemical, put it in a test tube, and then you can figure out where it lands in the frequency spectrum. And in that way, you can kind of calibrate where things should land. So we've got all these different chemicals, and we can measure them in the brain. Now, in the brain, things get more complicated because those chemicals get slowed down by their interaction with the different macromolecules. So you get the best signal when the chemicals are rotating very fast, like in pure water. So you put N-acetyl aspartate in pure water, it tumbles very fast, and you get a nice sharp peak. Now, in the brain, guess what? There's proteins, there's lipids, there's all kinds of stuff that slows it down, so you don't get as sharp a peak as you do in pure water. But we can still measure the chemicals in the brain, and I'll show you some examples. So here's again, here's lactate, and this is just from a pure state of lactate without brain. To see what it looks like. So at 1.3, so important, 1.3 ppm is where the methyl group of lactate lands, and then the methylene group lands up here, 4.1, 4.2 something. Okay, and then we use a technique called J-resolved because there's so many chemicals in the brain, they overlap. It's hard to see the forest through the trees because there's so many chemicals. So to weed out and try and get through all the forest to see just a couple of chemicals, we can use a J-resolve technique. And we use that technique in different ways. So here's a paper by Stephen Dager where he used MRS to investigate hyperventilation in subjects with panic disorder. And so we were able to measure the increase in lactate while they are hyperventilating. And so there's an increase of lactate in the brain as a function of time. So we were able to measure that during the scan. And then, now here is a paper by Kurt Weaver where we measured lactate in patients with mild cognitive impairment. Now, the reason why I really like this is because we're applying this lactate measurement to a clinical population. And not only that, we tested a new, well, it wasn't a new treatment. So Kurt, what was the treatment we tested?
Exercise.
Exercise. So it's so simple. Something as simple as exercise can really help the brain. So you're helping the brain by exercising. And so we were able to measure lactate in these patients during their different in the different phases. So anyway, I really appreciate these different co-authors here that helped us. So notice we have Rebecca Logston, Ellen McGow, Satoshi Minashiyama, Elizabeth Aylward, Natalia Kleinhans, Thomas Grabowski, Susan McCurry, and Linda Terry. So it takes a team of people. So you can never just do research in isolation. It takes a huge team of people with a lot of different expertise. And that's what we had here. Is we had a lot of different skills with different expertise. Okay, now I'm just going to say a few things about phosphorus. So Eric here and Kevin, they like to do phosphorus spectroscopy. So that's a different nuclei. So in the body, you've got plenty of carbon, you've got hydrogen, and you've got phosphorus. Now, the reason why phosphorus is important is because it's energetics. So in order to be alive, we have to have energy. And we need this currency of energy called ATP. So you need ATP. It's kind of like money. The more ATP you have, the more energy you have. So you need the ATP to live and to keep going. But we've done some studies with different MRS, and I've collaborated with Kevin and Eric on different studies. And one of them, we measured tumors. We did tumors, and we've done different techniques. We've done muscle. And here's an example of a phosphorus spectrum from a normal brain. Okay, and then if you take away the oxygen, now if you take away oxygen, see that tall PCR peak? It's going to be influenced. So if you take away oxygen, the PCR peak is going to come down because you need oxygen, you need metabolites to keep that PCR going. So you can't live very long without oxygen, okay? You probably only can go for about 6 minutes, whatever, because your PCR goes all the way down, you lose all your phosphoenergetics, it starts looking really ugly. But anyway, we can measure all these properties with the phosphorus spectrum involving energetics. Okay, so here's some of the projects that we use that involved MRS. We've done chemical brain imaging of autism. We've done visual deprivation, perinatal asphyxia, molecular imaging of cancer. And so we've done quite a bit with cancer. And then liver, we did a project with liver fat. And so the techniques that we used are single voxel, multi-voxel. There's spectral editing techniques in order to focus on different molecules. Now, here's a proton spectrum of the brain, which is isolated using PRESS. So see that little blue box? That blue box is a place in the brain that we can isolate and say, I want the spectrum from just the blue box, nowhere else. So you're getting a spectrum from just that box. Or we can do multi-voxel, which shows we can get several spectrum from across a couple of slices in the brain. So each of those boxes comes from a different place in the brain, and we've written software that allows us to make images out of this. We can take one of those peaks and we can make an image out of this using different software techniques. And then there's Megapress. Megapress is a technique to separate, for example, GABA. GABA is an important neurotransmitter in the brain. And but you cannot see GABA unless you do a special editing technique because it's buried under the large other chemicals that you get in the brain. So in order to see some of these chemicals, you have to use editing techniques. So here's the GABA after subtraction. Here's the creatine, and so you see the creatine kind of dominates things until you edit it, and then you're able to see GABA after the subtraction of the Megapress subtraction. And then here's the technique that we use with Stefan Posse, and we use this for many years in the GE scanner at the 1.5. It's called Pepsi. Pepsi's not a soft drink. It is a spectroscopy sequence. So it's proton echoplanar spectroscopic imaging, so Pepsi. And this is the Pepsi pulse sequence. So this was developed by Stefan Posse, but we are the ones that were able to program this into the GE scanner. So at the time we were doing this in the GE scanner. Took some programming to get all those pulses in there. Okay, I'm going to move on to functional MRI. How much time do I have?
We still have 35 minutes.
Okay,
You're
okay.
not even halfway.
Okay. So now I moved on to functional MRI. It's a technique for measuring brain activation during a mental task. Now, the beauty of functional MRI, it's completely non-invasive. You don't even need to inject a dye in order to make this happen. So the brain is scanned every 2 to 3 seconds while different stimuli conditions are given. So I'm going to give an example. So we studied dyslexia for many years. We studied children with dyslexia. And during the time they were in the scanner, we showed them a language task. Okay, so here's one of my favorite language tasks inside the scanner. The subjects are asked to indicate whether the pink letters in the top word could represent the same sound as the pink letters in the bottom word. So you see these funny words, float and drow? So those are non-words. They are non-words, but they are pronounceable words. They are pronounceable, but they're non-words. So that this person in the scanners has got this button, and they hit the button if the answer is yes. Can these two letters make the same sound or not? And so one of the conditions is to make this judgment of the pink letters, and then the control condition, you always need a control condition, are these sequence letters in part B here, are the two sequence of letters the same? So they're still looking at letters, they have to make a judgment, but it's a completely different part of the brain because the sound of letters is in a certain place in the brain. There's a part of the language brain where you can figure out the sounds of words and the sound of letters. That's called phonological processing, and that's in a very special part of the brain. When you're just looking at these letters and trying to decide if the same letters are the top and the bottom, that is in a different place in the brain. So we're able to do this on, off, where we can alternate back and forth probably about 5 or 6 times. And then we can get a brain activation map of where this occurs. And I'll show you a paper where we did that. But anyway, this causes a nice activation in a person who knows how to read. You can write. But a person with dyslexia, they have a harder time with this. With dyslexia, they have a hard time with phonological processing. And so their brain activation would appear in a different place. And the thing, so with Dr. Virginia Berninger, she was the brilliance behind the psychological education to transform these children. She would take our children after we did the scan, and she would give them this educational treatment that would last probably a couple of months. And by the time they got done and we re-scanned them, their brains had changed. And their brain activation had changed so that their activation was more normal for doing phonological processing. So their brains became more normal after this brilliant treatment by Dr. Berninger. Okay, so here's one of the papers that we did, functional brain and functional networking clusters. So Dan Peterson helped us with this one. Dan Peterson is exactly is right there. So the brilliance of this is that there's a toolbox called the Brain Network Toolbox, which allows us to figure out where the functional networks are in the brain. And so Dan was able to help us figure out how to use that toolbox to make these clustering coefficient in response to an instruction. So again, I always enjoy doing some kind of response to intervention in all of my projects. And so in this project we did several different levels of reading complexity. We would go to the single syllable, to the full word, to a phrase, to a full sentence. So we would add upon these complexities of language, and it's called multi-level reading brain. So each time you add another complexity to the brain has to work harder when you add a new complexity. So that was the purpose of this paper, was to measure these changes in response to instruction. Now, another project that I did with Hunter Hoffman was measuring functional MRI during pain-related brain activity. Okay, now pain. We had this hot plate that we would put on their foot. Does this sound fun? You put this hot plate on their foot, but we paid them a lot. We paid them a lot. Say, if you'll be in our study, you get to get this hot plate on your foot. We'll pay you a lot, but then you get to do this really fun thing called virtual reality. So virtual reality is another treatment. So if you're going to have pain, so before we did the virtual reality, all the pain centers would light up in the brain. So there's certain pain-related brain activity. You put it on and off every 30 seconds. So the temperature in the hot plate would go on and off, on and off. So during the time when there was no virtual reality, all your pain-related brain activity would light up. But then, if you're doing virtual reality at the same time, this is such a fun thing. It's called Snow World. And in the Snow World, you put on these virtual reality goggles, and you're going down the river, and these sea creatures fly up, and you're shooting snowballs at these sea creatures. Sound like fun? Sound like fun? And you have so much fun with this, you forget about your pain, and your pain goes away, and you don't have pain anymore. So that was the main result of this study, is showing that when you're doing virtual reality, the pain-related brain activity goes away because you're having such a great distraction with all those different sea creatures. Okay, now I'm going to move on to diffusion tensor imaging. So diffusion tensor imaging is a technique for measuring the structural properties of white matter and fiber tracts, nerve bundles in the brain. So before diffusion tensor imaging, the only way we could measure these nice fiber bundles was to take the brain out and stain it so we could see the axons and stuff. So in order to be more non-invasive, we can leave the brain inside the person. We can leave it, and we can measure these beautiful properties of these fiber tracts. So the brain is wonderful because it's like a network of telephone wires. It kind of looks like I'm going to show you some pictures. It kind of looks like a network of telephone wires connecting different parts of the brain. So the brain is scanned with several different diffusion gradients in order to allow reconstruction of tensor properties of the axonal fiber tracts. Okay, here's one paper we did with diffusion tensor imaging where we measured the repetitive blast exposure. These are veterans from Afghanistan. These are veterans from the VA hospital who were scanned with diffusion tensor imaging. But we also correlated their amount of blast exposure. Okay, so if you're a veteran, when you're in the service, these veterans were exposed to this heavy artillery. So every time this gun fires, it kind of shakes. It kind of shakes their head, and they would get brain damage because of when you fire the gun, it rattles. So even if you put headphones on, and you fire the gun, it just rattles your whole brain. So that's not good. And what we showed is that there's some cerebellar damage due to this exposure to this repetitive blast. Okay, now this is a fiber tract. These are fiber tracts. So my favorite fiber tract is right here in red. It is called the arcuate fasciculus, and it is used in language. So I see it kind of looks like a network of telephone, it looks like a network of wires. So the arcuate fasciculus connects Broca's area, which is speech production, to Wernicke's area, which is speech recognition. So it connects these two parts of the brain, helpful in language. And so we can measure these different structures using DTI, and we can isolate them depending on where we put a seed region and the connecting region. So the brain is a function of systems. You have a language system, you have a motor system. So in blue here we have the motor tracts coming all the way down to the spinal cord. So coming from the spinal cord all the way up to the motor cortex, we have the cortical spinal tracts. And then in green, it is called the inferior fronto-occipital tracts. This is involved in language processing, especially when vision involved. So back here is vision. So whenever you look at an object, the brain says, okay, I need to make sense out of this. When you're looking at an object, the brain has to make sense out of it. So it sends it different places to try and make sense out of what you're seeing. It could be a word, it could be a face, it could be some complex structure, but the brain always tries to make sense out of it. And in order to make sense, it has to send it to a higher order processing station in order to make sense out of what you're looking at. And so we did this study with a neuroimaging of the bilingual brain. So it turns out when you learn a second language, the brain undergoes another transformation. So you learn English, good, or if you learn a second language, the brain starts to rearrange and it says, okay, I'm going to put your first language in this part of the brain, and I'm going to put your second language right over here next to it. So there's been cases when people have had strokes where they wipe out one of their language, but not the other language. So sometimes you can keep one of your languages after you've had a stroke because it only wipes out part of the brain. So it's interesting how the brain says, okay, English is here and Spanish is over here. And then it goes through that process. But we did this study with different diffusion techniques measuring brain correlates of listening and speaking in the second language. So this is with the group I collaborate with, Patricia Kuhl from iLabs. So she has a group at iLabs. Here's another one of the papers we did with Ping Mamia from iLabs and measuring, and again, this was in diffusion in bilingual adults. And here is another diffusion we did with perinatal asphyxia. So Sunny Jewell, Dr. Jewell, is in the Department of Pediatrics, and she studies the effect of different perinatal asphyxia, or we also did a study with premature infants. So we were part of a multi-site project to study premature infants, where she wanted to test a new treatment to see if we can improve the brain. So one problem, when you have a premature infant, they are at risk for brain damage because they're not getting enough oxygen. So their poor little lungs are not fully developed when they're premature, and that means that they're not getting enough oxygen. The brain takes a hit. The brain takes a hit. They're not getting enough oxygen. So we were testing new treatments to help the brain in these cases where you're not getting enough oxygen. And now I come to another study that uses diffusion MRI. So Rebecca Petroff is a graduate student. Raise your hand. Rebecca, she's right back. She's a graduate student, and she's doing a study to study the effects of domoic acid on the brain. So domoic acid is kind of like a poison, right? It's a poison. So it kind of poisons the brain. So she's measuring the effect of this poison on the brain and seeing how it affects the diffusion diffusion connections in the brain. And so she's still working on this, and so she's doing a great job. She's learning all these different techniques for diffusion and other techniques in order to do it. And working with her is Tom Burbacher. Raise your hand. So he's back there. He's helping with this study. So we all have different expertise, and it takes a team of people to make projects like this work in order to understand the brain and how it works. Okay, so I enjoy the testing of new treatments for the brain. There is conventional medicine treatments, pharmaceuticals, and I've also done some testing of alternative medicine treatments. I'm going to show you some of the things I've done before. So whenever you test any wacky new treatment, it's good if you can use a scientific controlled method. So we were testing the effects. It's a pulsing magnetic field treatment for multiple sclerosis. So it's way out there. So most physicians, they think it's crazy. Okay, but if you can scientifically test it using good outcomes with a controlled population, then you can convince somebody that you might have an effect. So anyway, we're testing the effects of this pulsing magnetic field. So I like magnets. I like magnetic fields. So there are magnets in the MRI scanner, but there's also therapeutic magnets. So there's therapeutic magnets. And so we showed that there was an effect on the brain using this treatment. And we showed that it could be helpful. I mean, it's not a cure. It's not a cure, but it did help improve some of the symptoms when they went through this treatment. Now, another therapy, this is very alternative, another therapy that I investigated was that there was this Hawaiian healer. Okay, there was a Hawaiian healer, and we were doing a study of the effect of this Hawaiian healer on the functional MRI of certain parts of the brain. So this Hawaiian healer would stand outside the scanner. The patient laying in the scanner was inside the scanner, pulsing. We were pulsing away and scanning the brain, and we were measuring the effect of this healer on the person inside the scanner. And so we showed, with the 30 subjects that we could get an effect of this Hawaiian healer. So this is definitely alternative medicine. Okay, this is very alternative, but Western medicine doesn't have all the answers. We have Eastern medicine, we have acupuncture, we have several different kinds of medical techniques that could be helpful. And I have an open mind as long as it's scientifically tested. So if you're going to make a claim about something being helpful, you need to have a scientific basis. You need to show in a controlled way that it's going to be helpful. So anyway, I like investigating these different techniques. Okay, so now I'm coming to near the end here. My favorite, so since I'm retiring, I can have my favorites, okay? I have my favorites. My favorite indicators of brain health are 10 millimolar N-acetyl-aspartate as measured by MRS. So I've shown time and time again, if your N-acetyl-aspartate goes down, your brain's in trouble. So at a nice healthy level of 10 millimolar, you've got a healthy neuron. But if in some diseases like stroke, MS, Parkinson's, there's several diseases that'll bring Alzheimer's. In Alzheimer's, NAA goes down. So NAA it drops down, And then my other favorite is GABA. So 1 millimolar of GABA is good. So in autism, they don't have enough GABA, so it's too low. So there's diseases where the GABA is too low, and then there's diseases where the lactate is too high. So you want low lactate. You don't want a lot of lactate. You want a low level of lactate. So if you have a good blood flow to the brain, your brain gets enough blood in order to do glucose metabolism in the right way. So if you don't get enough oxygen, lactate comes up. So lactate is an indicator of badness. This is an indicator of badness. Okay? So the lactate, too high of lactate is an indicator. So anyway, and then my favorite neurostructure is arcuate fasciculus. So if you have strong fibers in that red fiber tract that I showed you, that's an indicator of brain health. And then your ability to form my favorite task, can the letters in pink make the same sound? If your brain can make this task and you can activate the right parts of the brain, I claim that's another indicator of brain health. So I think these are my favorite indicators of brain health. Now, I have to give you a recommendation. I have to give you a recommendation to promote brain health. So one of my favorite meals that I like that's good, healthy, coconut curry chicken with GABA rice. Okay, GABA is not only a neurotransmitter. You can find it in certain rice. You can get this at Central Market in Shoreline. That has a bag of GABA rice. Okay, GABA is one of my favorite chemicals. You gotta like that. So GABA is good. But now there's in India there is a village that has unusually low levels of Alzheimer's disease. So this is in India. So one of the potential factors is diet. And guess what they eat? They eat curry and rice. They eat curry. They eat garlic. I mean, there's certain diet things that could help your brain. So curry, garlic. So anyway, to promote brain health, I think there's several things we need to do. Some of them are diet. There's other things you can do with exercise, and you can also do things with how mentally active you. It's good to keep your brain actively solving problems. So one problem is after people retire, they think, oh, I'm done. I can just become a couch potato and I don't need to use my brain anymore. Bad, that's bad. It's bad because it's good to keep your brain active. It's good to keep it going, and that'll help your brain as well. So I would like to thank all my many collaborators and staff who've helped me over the years. I'd like to thank the wonderful technical support of different people who helped me with the scanner and make things work. And I'd also like to help all the radiology, financial administration staff for helping me with my grants. Thank you.
Thanks, Todd. That was really great. Does anybody have any comments or questions? I'll let you call on people.
Okay, go ahead, Tom.
How are you going to keep your brain active?
Well, I'm going to keep exercising. I have a few projects. So one of the projects I'd like to do is, so my brother is a musician, and so I like to do music with him. So I play the guitar. I like to do music. I think music is good for the brain, that kind of stuff. Okay, Tim?
I will have a gift for you. So it's from Dan and I. Now you know what this is, but you haven't seen it yet. So I'll give that to you. Sorry I didn't have time this morning to wrap it properly. It's just got a little bit of tape.
Did we get a hint?
It's a brain.
It's not just a.
It's
Okay.
not just
Okay, wait. So explain how you did this. How did you do this?
Well, I tried my best. I used every method I could find to try to locate a model of your brain, and I could not. So I had to ask you for that. So we got a model of your brain. We got a structural scan of your brain. Dan turned it into a 3D model, and then I 3D printed it. And full scale, which was quite a task. If I had looked more closely at the numbers.
So
before I
if my
started,
brain
yeah.
Your brain, bad brain health.
if my brain goes out, I can just plug this in. Plug this one in, right?
And it's a big brain too. I think if I'd have known when I started it, I would have scaled it to 90%. It took a long time to print.
Thank you so much.
Todd, comment and a question. Comment is like when I hear your contribution to science and your journey. It's really remarkable, like, the kind of work you have done, and it's what a great contribution. So certainly thank you for your services, and the future generation you have inspired, people you have mentored will be an enduring legacy in this place. And so that is the comment. The question is, I look at your faculty and the scientists and others, and I'm thinking your recruitment strategy was all those you listed as the criteria for brain health. Maybe you were imaging them before you recruited them.
I was trying to image it. I was trying to figure it out. That's right. That's right.
You're giving me ideas.
Thank you. And you're from India, right?
Yes.
India. So India has this village. Well, they have this village with very low Alzheimer's.
I won't recruit from there though. But maybe GABA rice next time for lunch.
We're having it as our catered. There you go.
Yes, thank you.
Good
Thank
luck in
you.
your retirement, Todd.
Okay.
Yeah.
You'll be missed.
All right, thank you.
Thank you. Thanks, everyone. Let's do this thing.
So the two halves.
That was on the inside.
You give me your fork.
Oh, yeah.
I opened up my
Good luck.
combo piece. There was no fork in there. $2.
Yes, I can still do
I think oh, you should just trying to stump people in
Let them know. Peace of mind.
Absolutely.
So many familiar names. You know, I